So last time I have told you what is the botanical origin and what is the progenitor species. So this time we see the progenitor species of these crops. Among all the crucifers, the cabbage, cauliflower, broccoli, Brussels sprout, all these crucifers belongs to the genus Brassica. And among all these crucifers, kale was the crop first originated. So kale and leafy non-heady cabbage, they were originated from Brassica vulgaria variety silvestris. This is the primitive kale. From kale and non-leafy heady cabbage, but originated from the primitive kale Brassica vulgaria variety silvestris. From this leafy non-heading cabbage or head cabbage was developed. Whatever we are seeing in the market nowadays, the cabbage that forms the head, that is head cabbage that was originated from leafy non-heading cabbage. And Savoy cabbage. Savoy cabbage means where the leaves have curls. So the Savoy cabbage, it was originated from Brassica vulgaria variety costata, the common name Portuguese kale. And broccoli and Chinese kale were originated from Brassica critica subspecies Nivea in Italy. Broccoli was originated in Italy from Brassica critica subspecies Nivea. Cauliflower was originated from broccoli. Generally, we know much about cauliflower, less about broccoli. Into India, cauliflower was introduced earlier and broccoli was introduced much later. So we think that broccoli is the new crop. No, in domestication or in origination, broccoli was originated earlier and cauliflower was originated from broccoli. And Brussels sprout was originated from Brassica vulgaria variety Ramosa, common name is thousand head kale. And Mulcol was originated from Brassica vulgaria variety Medulosa, narrow stem kale. And onion was originated from Ilium barbilobi. Similarly, garlic was originated from Ilium longicausis. So carrot from Queen Anne's lace, Dacus carota species carota. And the Asiatic radish originated from Raphanus sativus variety Raphanus troides. Beetroot from Beta vulgaris subspecies maritima. And the grain amaranthus from Amaranthus hybridus. Spinach from Spinacia tetranda. Lettuce from Lactica cereola. Potato. Last time I told you some confusion was there about the ploidy of the potato and the sweet potato. Potato Solanum tuberosum is autopolyploid, it is autotetraploid. But another cultivated tetraploid potato, Solanum stoloniferum, that was allotetraploid. Last time the confusion was there in last lecture. So tapioca was originated from Manihot esculenta subspecies Lamellicoria, and sweet potato was originated from Ipomea trifida. Still there is a lot of confusion about the origin of sweet potato, whether it has come directly from the Ipomea trifida or it might have come by the cross between Ipomea trifida and some unknown species. So if we see the important crops in the cruciferous family, cabbage, cauliflower, mulcol, broccoli, Brussels sprout, kale, Chinese cabbage and Chinese kale. You please remember the botanical names. So this is generally called U triangle. This clearly explains how different species of Brassica were originated. So this is the our Brassica vulgaria. All our vegetables belong to this group Brassica vulgaria. There the haploid chromosome number is equal to 9 and the genome is CC. So first there were Brassica rapa, Brassica nigra and Brassica vulgaria. From Brassica rapa and Brassica vulgaria, natural hybridization, Brassica napus formed, and from Brassica vulgaria and Brassica nigra, Brassica carinata was formed, and from Brassica nigra and Brassica rapa, Brassica gentiana was formed. This is called U triangle. This explains how different species of Brassica were originated. So the geographical origin. If you see, almost all these pole crops were originated in the Europe near Mediterranean region. Cabbage in eastern Mediterranean region, cauliflower from Syria, broccoli from Italy, Brussels sprout from Belgium, and mulcol and kale from Mediterranean region. Chinese kale and Chinese cabbage were originated in China. But these pole crops were migrated to China by humans, and there they were modified and into Chinese kale and Chinese cabbage. So broccoli was much introduced into India later in 20th century. Though it is an ancient crop, though broccoli was the progenitor of cauliflower, To India, it was introduced much later in 20th century, in 1990s. So we see the cabbage. Cabbage is a largely cultivated crucifer vegetable, and it has an anti-cancer property due to the presence of indole 3 carbinol. And then one fermented product, popular product from cabbage, sauerkraut. Generally, the seamen, when they go to fishing into the ocean, they take this sauerkraut and they eat it. Fermented product, so we can store it long time. It can be used against scurvy. Scurvy is due to the deficiency of vitamin C. So against that it can be used. And based on the color of the cabbage head, whether white color, red color, or savoy color, different forms are there. That's called the variety capitata, form a alba, white color head, form a rubra, red color head, and variety saboda is the savoy cabbage. Savoy means the leaves have pulse. So cabbage is a cross-pollinator. All crucifers are cross-pollinated. That is due to the presence of protogamy. Protogamy means the female pods come to mature earlier than the male pods. So the gynoecium matures earlier than the androecium. This cross-pollination is due to protogamy, and it also has self-incompatibility. Self-incompatibility means self-pollination may not occur. The pollen of the same plant cannot fertilize the ovules of the same plant. So that self-incompatibility type is sporophytic here. That means the self-incompatibility is governed by the somatic chromosome number of the parent. So the inflorescence of cabbage is catkin. This is very important because this is the unique inflorescence among all the crucifers. Generally, crucifers have either cymose or racemose, but the catkin is having the the cabbage is having the catkin inflorescence. This is a very unique one. Catkin means the inflorescence is drooping type. It droops. And it means it is hanging type groups. So the flavor of the cabbage is due to presence of the glucoside synecrin and 2-propenyl isocyanate. And there is a male sterile system in cabbage for hybrid seed production. It is cytoplasmic male sterility. The source of cytoplasmic male sterility is from Brassica nigra, and it is an Anand cytoplasm. Actually, originally it is from Brassica town 40. Brassica town 42, the cytoplasmic male sterility was transferred to Brassica rapa. From Brassica rapa, it was transferred to cabbage. And based on the head shape, there are 3 types. Head shape generally we can see as P by E. P means the diameter of the polar, that means from top to bottom, and equatorial diameter, that means horizontal length. So the vertical length and horizontal length, the ratio of the vertical length and horizontal length, it gives us the shape of the cabbage head. If the P by E, polar diameter by equatorial diameter ratio is 0.8 to 1 means it is a round head or spherical head. If it is 0.5 to 0.6 means it is drum head or flat head. If it is greater than 1 means it is conical head. And the major pest of the cabbage is diamond black moth, Rutella gynostella. Why it is called diamond black moth? If you see the adult moth back side, there is a diamond moth. So it is called diamond black moth. So there is a classification of varieties in cabbage, early, mid, and late based on the time of sowing. And based on the head type, they are again classified into round, flat, and conical. So you can refer any textbook for the classification of these varieties. I have not given here. And if cabbage is grown on the saline soils, it is prone to black leg disease. Black leg disease is caused by fungus, Homa lingam. The breeding methods followed in cabbage were mass selection, recurrent selection, family breeding. Family breeding especially followed in pole crops. In pole crops, family breeding is followed. It is similar to the line breeding, but the selection goes for more than 1 generation. The family breeding is generally followed in pole crops. So nowadays hybrids are very popular, though less number of hybrids were developed by the public sectors, but a huge number of cabbage hybrids were developed and marketed by the private sector in India. And synthetics are another type of varieties developed in the cross-pollinated crops, especially crucifers among the vegetables. Generally, synthetics and composite varieties are developed in cross-pollinated crops. We take inbred lines and we allow the open pollination among the inbred lines, and we will collect equal number of seeds from each parent. We will mix them, we will grow them. This is a simple way of developing a synthetic seed. So you can refer the textbook for the detailed procedure of the developing a synthetic variety. So the ideal number of inbreds for developing a synthetic variety are 4 to 6. This is a previous question. This 4 to 6, you can remember this figure. Ideal number of inbreds for synthetics development 4 to 6. So the synthetics are based on the exploiting the general combining ability and the additive genetic variation, whereas hybrids are based on the specific combining ability and non-additive genetic variation. You just refer in the genetics book what is the additive and non-additive variation. So the resistance to powdery mildew is governed by single dominant gene and the resistance to Fusarium yellow. There is a two types of resistance in cabbage. The type A resistance was governed by a single dominant gene, whereas the type B resistance was polygenic, but it breaks at 22 degrees centigrade. It is temperature sensitive. So for developing the inbred lines in cross-pollinated crops, we have to do selfing. So up to 6 to 7 generations of selfing in a cross-pollinated crop will give us an inbred, which is almost 97 to 98% of homogeneity. So there is self-incompatibility. So we cannot do selfing in cabbage. So for developing the inbred lines in pole crops, especially in cabbage, cauliflower. So how for development of the hybrids we need inbred lines because the hybrid should have homogeneity. So to ensure the homogeneity in the hybrid, the parent should be homozygous. But the cross-pollinated crops are heterozygous. So we have to convert the cross-pollinated crops into homozygous form. How we get the homozygous form is by selfing for 6 to 7 generations. The homozygous form of a cross-pollinated crop is known as inbred. So if there is self-incompatibility in cabbage, so how do we do selfing and how do we get the inbred in cabbage? So to overcome the self-incompatibility, there are three methods. Bud pollination. Bud pollination means before anthesis, before opening of the flower, we have to do pollination because the self-incompatibility alleles become active after the flowering. When the flowering occurs, the self-incompatibility will come into operation. Before flowering, there is the self-incompatibility alleles will not function. So before the flowering, that means we at the bud stage, 2 to 3 days before anthesis, we have to do the pollination. Otherwise, we can spray 3% sodium chloride 1.2 hours to 1 hour after the pollination, or we can expose the stigmas to 3 to 5% CO2 for 8 to 24 hours at 100% relative humidity. These three methods can be followed to overcome the self-incompatibility in cabbage. So there are some important varieties like Posa agathis, the tropical cabbage, set seeds in subtropics. And the tropical varieties do not require vernalization. They can set seeds in the lines. And Posa sambandh is a synthetic variety. It is suitable for the high density planting because of the short stalk length. And Posa mukta is resistant to black rot. The black rot was caused by a bacteria, Xanthomonas campestris. And Posa drumhead variety was resistant to black leg, which was caused by the fungus. And there is a Savoy cabbage variety, chieftain. So the next important crucifer is cauliflower. Cornish cauliflowers were first introduced to India. Cornish cauliflowers were England. So the Cornish cauliflowers of England were first introduced to India. Afterwards, other European types were introduced to India. Cornish, Roscoff, Italians, and Northlands. All these were temperate types. So how the tropical Indian cauliflowers originated? After bringing them here, there was a natural crossing among these European types and the selection by the farmers for the heat tolerance and suitability for the adaptability for our Indian conditions. So by natural crossing and selection by the farmers, our tropical cauliflower was originated from the European types, especially the Cornish one. So this tropical Indian cauliflower is having stable self-incompatibility, whereas the European cauliflowers were the thermosensitive because at high temperatures their self-incompatibility will be broken. So the inflorescence of cauliflower is racemose, whereas the inflorescence of cabbage is catkin and the cauliflower inflorescence is racemose. So in Indian cauliflower generally we prefer the semi-erect types than the erect types. So into cauliflower, male sterility was transferred into cauliflower through a recombinant DNA technology. The Barnes gene governs the male sterility. The Barnes gene was isolated from the bacteria Bacillus amyloliquefaciens and it was transferred to the Barnes gene, whereas the Bar gene is the herbicide tolerant marker gene and the Foster is the restorer gene. like cabbage, cauliflower, CMS was transferred to cauliflower also. The CMS source in case of cauliflower is Ogura from Raphanus and the same Anand from Brassica rapa. So different breeding methods followed in cauliflower were mass selection, recurrent selection, back crossing, and hybrids. Recurrent selection was used to develop the synthetic varieties. So here also, to overcome the self-incompatibility, we have to do the bud pollination. And one more important thing is the orange card color. Orange card color in cauliflower is found by a single dominant gene. This was the can somebody mute? The orange card color was found by a single dominant gene. This was the previous question. The OR, the orange card color in cauliflower was found by the single dominant gene, and the black rot resistance is polygenic dominant. And there is an important classification of varieties based on the temperature requirement. The cauliflower varieties are classified into 5 types: extra early, early, mid late, and last late snowball types. So you refer any textbook, get the classification of these varieties based on the temperature requirement, and there are some varieties resistant to various diseases. And Posa megana is a tropical cauliflower. It can tolerate high temperature. And saline soils, growing of cauliflower on saline soils will cause black rot, and if you grow on acidic soils, it causes whiptile, clubroot, and white rust. So the tropical cauliflower varieties are Pusa Katki, Akka Kamti, Akka Vimal, and Akka Spurti. They set seeds in tropics. Last time, Ms. Toko, I think she asked me a question whether the tropical cauliflower needs vernalization. No, that tropical cauliflower do not require vernalization for flowering, but the snowball type, the late flowering snowball types require vernalization for flowering. So other crucifers, broccoli, Brussels sprout, and null they have anti-cancer property in the presence of isothiocyanates and indoles. And the broccoli has the sulforaphane compounds which reduce the risk of cancer. So inflorescence of broccoli is cymose, Brussels sprout, null racemose. Brussels sprout, null cauliflower, the inflorescence is racemose, whereas the broccoli it is cymose, and cabbage it is catkin. So among all the crucifers, broccoli is the highly cross-pollinated crop, 95%, followed by null, followed by cabbage, and followed by Brussels sprout. And there is a 6 male genes are identified in broccoli. Again, the CMS source is Brassica nigra or Ogura of Raphanus. So if you see the edible portion of the broccoli is immature inflorescence, similar to cauliflower. The Brussels sprout, the axillary buds of the Brussels sprout develop into small heads. So it is called as a mini cabbage also. The axillary buds are known as sprouts and buttons. And the null the edible portion is knob. The swollen stem above the base of the stem, it swells and it forms a knob-like structure. So this is the edible portion in case of null pole. So in case of Brussels sprout, the axillary buds turns into swollen heads or swollen sprouts. So topping is an important intercultural operation in Brussels sprout. Topping means removal of the apical portion. So the removal of the apical portion allow the axillary buds to grow into bigger sprouts or big heads. So topping, removal of the top growing point or apical portion is an important intercultural operation in Brussels sprout. So it also useful for the mechanical harvesting also. So the null pole is harvested when it gets the 6 to 8 centimeter diameter. And kale, when comes to kale, there are 2 types of kale. One is Scotch kale with much curd leaves. There is Siberian kale with less curd, blue purplish leaves. 2 types of kale. One is Scotch type, one is Siberian type. And Chinese cabbage are also 2 types. Pasai, it is having elongated head like horse lettuce, and pop chai is having no heading rosette leaves. You see the botanical names are different. Pasai and the pop chai. Just see the difference in the botanical names also. So there is some intergenetic hybridization was made among the crucifers vegetables, and some vegetables were developed by hybridizing the different genera of these crucifers. They are known as Raphanus. You all know that radish and cabbage. It was a failure. The breeder was expected to get the roots of radish and the heads of cabbage, but unfortunately he got the reverse roots of cabbage and top portion of radish. So Raphanus and Nabicol. Nabicol was a cross between kale and turnip. Calico between cauliflower and cabbage. Rutabaga between turnip and null. Hakuran between cabbage. Hakuran is the previous question. Hakuran, cabbage is Chinese cabbage. It is resistant to soft rot and it is tolerant to heat also. Hakuran was previously asked, and Rutabaga also previously asked.